Hi everyone. Just wait just a few minutes before coming. 1 minute. Okay, I think we can start. So, hi everyone. Welcome to our first webinar of 2026. My name is Luciana Lima. I'm a PhD student at UNIRIO in Brazil, and together with my colleague Breanna, who is a postdoctoral researcher at the University of Manchester in the UK. We are very happy to be your hosts for this session. And for the first time, we have three talks in the webinar. And now Breanna will introduce our first speaker. So thank you for coming and joining us.
Thanks, Luciana. So first up, we have Professor Masanori Arita, who has a PhD in information science and a professor at the National Institute of Genetics in Japan. He was a developer of the original mass bank database and was a supervisor of the MS-DIAL software platform. When the Metabolomics Society was established in 2005, there were no open repositories for mass spectra. MassBank first started as a database for standard MS spectra. Now, after 20 years, there are multiple repositories and databases, and efforts should focus on standardizing metadata description and on nomenclature to optimize the use of already existing data. His talk is titled From Sharing to Reusing Metabolomics Data. So over to you, Masanori.
Okay, thank you for your introduction. Let me share my slide. Can you see my screen?
Yeah, it's perfect.
Okay, then let's start. My title is From Sharing to Reusing Metabolomics Data. My affiliation is primarily National Institute of Genetics, but I also am affiliated with RIKEN Center for Sustainable Resource Science. And this research center was formerly called RIKEN PSC Plant Science Center, doing plant metabolomics much. Today, I'd like to talk about our history in metabolomics. As introduced, I joined the first society meeting in 2005, and this first meeting was held in Tsuruoka City, Japan. And for those who do not know much about Tsuruoka, here's the map of Japan. And Tsuruoka is north of Tokyo, so you have to fly from Tokyo by 1 hour. And it's a small city of population around 100,000. And of course, it's a local city. And 20 years ago, this city had no flavor of science, but Keio University took an eye on this wonderful city and set up an institute focusing on metabolomics. 20 years ago, it had a really good side. So Keio University ran big group in metabolomics, and I joined this group as the first developing team of mass bank project. And of course, there were no spectral databases for free, no open software repositories, of course, no Zoom, no WebEx. So the situation, research environment was quite different then. And at that time, many metabolomics were done on already using ESI, electrospray ionization, but there were other ionization schemes like APCI and others. And I believe platforms were also diverse, quite diverse at that time already. So when we started the mass bank project, my first job was to create a spectral search algorithm. The official project started in 2006, led by Professor Emeritus now, Professor Nishioka. And the main developer was me and Dr. Horai at that time. So I was actually the first programmer who wrote the mass bank, uh, uh, at that time, mass bank program for spectral searching. And then there were no good search algorithm for ESI mass spectra. So what I studied most was the spectral search algorithm for GCMS, especially the NIST search algorithm. I, I hope you all know about NIST library. And it was primarily for GCMS at that time, but it started to accumulate other mass spectra. So I extensively studied the NIST library search and created the first version of spectral search program. Of course, ESI fragmentation and EI fragmentation in GCMS is quite different. So yeah, what I thought uh, was to devise a good evaluation scheme for spectral similarity. For your, yeah, to explain the pictures, this is the very first version of MassBank website in 2006. And at that time, only two research institutes provided us free spectra, one from Keio University, the other set from RIKEN PSC Plant Science Center. So these were the only research institutes who kindly provided us so many spectra for free. But soon, many institutes followed. So we actually traveled around Japan to collect mass spectra that we can provide for free for the world. And as I said, fragmentation was a very difficult problem, and it is still a difficult problem, I think. Most of the software programs use dot product or cosine correlation to see spectral difference, but because spectra are so different according to fragmentation scheme, the calculation method is different between EI, ESI, and APCI, and other ionization. And my first task was to find an optimal parameter for peak weighting for spectral algorithm. So if you are interested in spectral match algorithms, I really recommend you to read this old pivotal paper written by Stein and Scott in 1994, and the Journal of ASMS. This work is still informative for you who are interested in spectral matching. And after establishing or after fixing the spectral search method, we yes collected as many spectra as possible from all over Japan, and then we extended our collaboration overseas. And since then, we have been acquainted with Stephen Neumann, IPB Halle, and Emma Szymanski, Airbag, Switzerland. No, not now Airbag. I'm sorry. She's now in Liechtenstein or somewhere in another place in Europe. Sorry, but they helped us quite a lot through collaboration to establish our mass bank consortium. And Massbank is now based one in Europe, one in the United States, one in Japan. But at that time, we had even more servers. Actually, at its height, at its peak period, we had more than 10 servers worldwide, not only Germany, China, but we had a server also in Brazil once. And these servers were federated, meaning each server has its own content locally. And when you query one spectra from one node, the query was distributed to all the nodes worldwide and collected all replies. So, as you can imagine, it took some time to see the full list, but it was really how federated consortium, literally. But this scheme was later abolished because of the cost for server maintenance and because internet security became a big problem. Well, anyway, this mass bank project kind of shaped the trend of uh, opening data or sharing data for free. And this time coincided with opening other category of metabolomics information, especially from HMDB in Canada. David Wishart started his program through Genome Canada very strongly, and he's now an established figure in many databases and also software programs. So we together advocated free distribution of mass spectra and also program codes. But as you can imagine, problems are quite similar still now, such as licensing or spectral quality, and also maintenance and securities. As for licensing, we took Creative Commons and we allowed users and also contributors to attach any type of Creative Commons at first. So some people put um, how non-commercial license or even stricter license in addition to Creative Commons. But with time, many spectra or almost all spectra were migrated toward free style, such as CC0 and also CC BY. So nowadays, you do not see many spectra under strict licenses, but we had, yes, big difficulty from requests such as retraction and no modification of spectra. For us, no modification or non-commercial was quite problematic, but as a repository, we received all kinds of spectra from all over the world. And after collecting spectra, now we could move into establishing software programs. And most well known are MS Dial and MS Finder. These two programs were both developed by Dr. Tsugawa. Now he's professor, but he joined my research team after getting his master's degree. So I supervised his PhD and he stayed in my team after getting a PhD. And these two wonderful programs were the fruit of international collaboration, especially with Professor Oliver Fein at UC Davis. And between year 2011 and 16, as you can see, we had a really wonderful program, wonderful support from JST in Japan and NSF in the United States titled Metabolomics for a Low Carbon Society. So with Professor Fein, we investigated many algal species, algal lipids under this program, including Nanochloropsis and also Euglena. It's not an algae anyway, but it's a microorganism producing very interesting lipids. And through this analysis, we developed not only MS Dial, MS Finder, but also splash code, spectral hash code for identifying the identity of mass spectra. If you do not know about splash code, please visit HMDB, and for each metabolite, you see the splash code associated with its spectra. So we developed many useful tools through our collaboration. So for young investigators, my real recommendation is how cultural difference is a really is really the key for creating synergy. Without collaboration with Oliver, I couldn't establish such wonderful team and probably no such good results in metabolomics. And this establishment led us to lipidomics. Lipidomics is a subfield of metabolomics. But as you know, in lipidomics, we use many theoretical libraries. So because lipids are the conjugation of several types of basic components like fatty acids or glycerol backbone and also small derivatization through hexoses and acid fatty chains, uh, through combining their fragment spectra, we can estimate or we can extend the variation of lipid classification and lipid structures. So in MS Dial, now you can find more than 30 lipid subclasses in its lipid library, lipid theoretical library. And this achievement was the result of very successful collaboration with Professor Makoto Arita, another famous Arita in metabolomics field. And uh, with this collaboration, we could establish many spectral libraries, especially for new lipids. For example, through the collaboration, we kind of discovered new lipid classes such as acylated sphingomyelin. Uh, Sphingomyelin is quite famous. It's the major component in our brains. But sphingomyelin can be acylated. And this acylated version is mainly abundant only in kidney. The reason is still unknown. Another strange lipid is, for example, very long unsaturated fatty acid in testis only. And the reason is unknown. Very long fatty acids have been known to be expressed in the eyes, actually, especially in cows and other bovine species, but we never expected even longer polyunsaturated fatty acids in mouse testes. So such new discoveries have been made in this untargeted lipidomics, but unfortunately, we still do not know why they are, for example, accumulated in particular organs like testes or kidney. So, so many biological questions have been left unsolved. So we expect young researchers to find out why biologically such very unique and interesting lipid classes are mm, accumulated or concentrated. And after this lipidomics library, my focus moved on to a repository for mass spectral raw data because now sharing and reusing standard reference spectra are on the track. But relatively, spectral raw data are not shared or not reused. And reanalysis is quite difficult still now. So my hope is that situation or environment where we can reanalyze other people's data quite easily through free software programs or free visualization programs. How can we do? we already have several spectral raw data repositories like Metabolites at DBI, Metabolomics Workbench in San Diego, or Massive. But although these platforms provide very useful interfaces for searching raw spectra, in-depth analysis is far more difficult. So to make such analysis more easy, my solution is to add much more detailed metadata for each raw mass spectral raw data. I don't know this is the right direction, but for this purpose, we established Metabobank in Japan, which followed the metabolite annotation uh, in a way that metabolites does. Metabolites, I think, is a really good repository with very rich metadata, but because of this, annotation takes time. It's really difficult to annotate all submitted information. So we try to support metabolites by creating a similar deep annotated raw data from Japan. So we are exchanging metadata with metabolites, and we try to attach as much information as possible through our two curators in my institute right now. And another how advantage of our Metabobank repository is it is linked with other omics data like next generation sequences and also gene expression data. When you submit genome information or functional genome information, you usually use DDBJ, GenBank, or ENA repositories, and these repositories asks you to attach metadata called bioproject and biosample. Bioproject for research projects, biosample for each sample extraction or sample preparation. And these metadata are actually common with metabolomics. So at Metabobank, we ask users to submit bioproject and biosample metadata to link metabolomics data with other omics information. And I hope this trend will influence other repositories so that we can connect different type of omics information more easily. And another important point of Metabobank is metabolite assignment file or MAF file. This is common to metabolites repository, but this type of metabolite annotation is more useful for other users or visitors to check what kind of metabolites are included and also for comparing metabolite concentrations or metabolite profiles. Or for data submitters, of course, this is a huge task, but for data reuse, this kind of information is essential. And of course, machine readable format is better, but for standard submitters, XML or other formats are hard to submit, so we chose simple Excel format for describing metabolite information. But nowadays, you can use AI, so translation from Excel file to other data format became much easier. So I think our choice of using Excel for metabolite annotation is feasible or viable now. What to do from now? We need to use AI as much as possible because it's really handy. And through AI, we can change in data format quite easily. So we need to be acquainted, or we need to be fluent in AI assistance. And coordination between repository is also essential because um, maybe different repositories share the same raw data information. So in such a case, statistics, taking or obtaining statistics will become difficult. So repositories need to coordinate more, but more than everything, I recommend you to check the website of Metabolites at EPI. It now collects more than 2,000 studies with detailed metabolite information. So for beginners and for experts, such detailed metadata is quite useful to view to see the overview of metabolomics field. In other words, data reuse is the key for the next generation. Now I'm coming to my conclusion. Maybe I'm going too fast, but anyway, my emphasis for young researchers is metabolomics is now becoming possible without measurement. I mean, from already measured data, you can do metabolomics. And when you do this, traceability of not only data, but also programs and your way of thinking is quite important. And you need to leave such traces in your research papers or in your resubmitted information to repositories. So from my view, from my experience of 20 years, what is most important is linking, not only linking data, but also linking people across countries and across research disciplines. And from my own experience, connection to very different researchers in different countries was a real, yes, benefit throughout my research career. So please go abroad, visit different laboratories, communicate with other researchers in different countries. That's my last word as conclusion. Thank you very much. And maybe I can take questions, then switch to the next speaker.
Thank you, Professor, for your talk. It was incredible. And I totally agree with this slide and conclusion. I think it's the future of the metabolomics and lipidomics. I mean, and we have a question in the Q&A box. How do we ensure spectral quality? Is this something the data owner should prioritize, or is it the database owner's responsibility to enforce?
Sorry, I don't see the question. Can I see the question or?
If you go down to the bottom of your screen, the Q&A box, it should appear there.
Well,
Can you
there's
see?
a question and answer box, but is it in the chat window? Oh no.
The question and answer box at the bottom of your screen
But
next to the show.
I see only one question. How do we ensure spectral quality? Oh, okay, this question. I'm sorry. Sorry, sir.
This
Now
question.
I no. Okay, okay. Well, it really depends. If you can find similar spectra in different places, of course you can compare. But if you have only one spectra for one very rare metabolite, there's no way. But if you can use how structural prediction tool like MS Finder or others, maybe you can check its quality more or less. But in my opinion, such spectral prediction programs are not yet mature. So yeah, it's very difficult to see the quality only from one spectrum.
I guess leading on from that, how do you see the balance in like in silico annotation tools and experimentally validated spectral libraries evolving over the next, say, 5, 10 years?
Well, for basic metabolites, we already have very good spectral libraries, but the problem is a library for rare metabolites or not well investigated metabolites. Probably the second talk by Shohei is the right answer. Sometimes we need to, yes, guess its spectrum or its retention time. So prediction is still very much important.
Thank you. Does anyone else have any questions? There's one here. Can we connect this type of database to uncover the dark metabolome?
Well, I'm sorry, I do not know what type of metabolites you mentioned by dark metabolome, but I think we still have many unknown metabolite classes, especially unknown derivatives and unknown mixtures. And most importantly, we do not know much about how peptides. Yeah, probably, yeah. Again, the second talk by Shohei will describe about such not well investigated peptides. So,
Great. Any other questions? Otherwise, we might move on to the next speaker. Okay, well, thank you very much, Masanori. And now
thank you.
we have Dr. Shohei Nakamukai, who has a PhD in agricultural science and is a postdoctoral researcher at the RIKEN Center for Sustainable Resource Science, Japan. He's interested in marine metabolomics and works on the identification and synthesis of bioactive peptides. And he will present on retention order prediction of peptides containing known non-proteinogenic amino acids. Over to you, Shohei. Thank you.
Thank you for kind introduction. Can I share my slide? Okay. Yeah. So, sorry. Yeah. Yeah. My name is Shohei Nakamukai, PhD, in RIKEN CSRS. And I'm in the Metabolomics research team. So, and my background is computational metabolomics, especially SDMS data analysis. And my research focus is identification of marine natural products. I'm using the machine learning-based retention order prediction. And my current interest is especially in peptides containing non-proteinogenic amino acid. We call them PNPA in marine sponges. So today, I'd like to talk about how retention of the prediction can support the identification of marine peptide. So, okay. So, sponge, Phyllum polyphera, branched off early in animal evolution. They inhabit a wide range of marine environments, from shallow reef to the deep sea. And they are filter feeders, removing particles from seawater. And it is known that the sponge contains a large number of chemical compounds. And these compounds have unique structures compared to those found in plants. So, furthermore, as shown in this graph, so the largest number of compounds has been found in sponges among marine organisms. Here is a sponge. So therefore, they are considered promising resources for drug discovery. So a major reason why sponges produce so many compounds is chemical defense. In fact, so only a limited number of organisms, such as sea slugs and some mollusks, are able to feed on sponges. And in many cases, so the true producers of these defensive compounds are symbiotic microorganisms. And diverse symbiotic microorganisms produce sponge compounds, but most producers remain unknown. And among the diverse compounds found in marine sponges, and peptides have attracted particular attention, especially so peptides containing non-proteogenic amino acids, we call them PNPAs, are characteristic compounds found in marine sponges. PNPA contains many non-proteogenic amino acids. Sorry. So, for example, the daptomycin, a clinically used PNPA, includes multiple D amino acids and ornithine and modified amino acids and ester linkage and fatty chain in its structure. And these structural features are considered crucial for their biological activity. And some PNPAs are known to exhibit unique mechanisms of action. For example, polycyanamide is a giant linear peptide found in marine sponges. It forms a helical conformation and creates ion channel in cell membranes, leading to strong cytotoxic effects. Because of such unique activities, PNPA are regarded as promising drug candidates with novel mechanisms. So, a common approach to identify natural products is to compare experimental MSMS spectra with reference spectra in public database. And this approach works well when the database contains main reference spectra. However, for marine natural products, the MSMS reference spectra is still limited in current database. Therefore, it is difficult to identify marine natural products through this method. And this is the reason why research in the marine natural product is not progressing. Therefore, in recent years, some studies reported the utilization of retention time in LCMS for the compound identification. Retention time reflects the interaction between a compound and the chromatographic column, providing useful information for compound identification. However, even for the same compounds, the retention time varies depending on the measurement conditions, such as the solution, column, and flow rate. So, therefore, Dr. Bush focused on the retention order, which is the order compound eluted from the column. If the reverse phase condition of the column are the same, the retention order does not change significantly. So it is possible to compare between different datasets. So in a previous study, they successfully predicted the retention order of small molecules by applying machine learning rank SVM trained on dataset and achieved a prediction accuracy of around 0.9. So in my study, I tried to identify the PNPA by predicting the retention order. Because the retention order is largely conserved across different measurement conditions. The dataset acquired under different LCMS conditions can be used together as training data. And to compensate for the limited availability of PNPA data, we utilized both peptides and small molecule dataset for model training. The peptide dataset supported the learning of amino acid moieties, while the small molecule dataset contributed the learning of modified parts. And our goal was to build a model that accurately predicts the retention position of PNPA relative to reference compounds. So, as training data, we utilized 4 small molecule datasets from platelet database and 5 peptide datasets using a previous study. And as test data, we measured 9 standard PNPA and 22 reference compounds under the same LCMS condition. These 22 reference compounds consisted of standard small molecule with varied retention times. Therefore, these reference compounds were used to evaluate the relative retention position of PNPA. And we use the rank SVM as a training model, which is suitable for learning retention order rather than retention time. First, the compound pairs were selected within each dataset. And the fingerprint, which is a method to express the structure of compounds. The fingerprints were used to express the compound structures. Using fingerprint as the input variables and the retention order as output labels, we perform the training on all compound pairs. And against the training datasets, first, to reduce the multicollinearity, principal component analysis, PCA, was applied to the fingerprint input data. Followed by fingerprint optimizations and model training, and the training model was evaluated by comparing the predicted retention order of PNPA standards with the actual retention order related to reference compounds. So these are the results of the retention order prediction. And when using only peptide or only small molecules, the prediction accuracy achieved 0.85 and 0.81 respectively. In contrast, so integrating both datasets for peptide and small molecule improves the prediction accuracy to 0.904. This result suggests that the combining peptides and small molecule dataset could capture the complex structural characteristics of PNPAs more effectively. So here, I demonstrate how our model can be applied to real samples. First, please prepare the prediction model from the GitHub in this link. And next, perform the LCMS measurement of a sponge extract together with a set of reference compounds under the same conditions. So in this example, we use a sponge sample, the species Breanna, collected in Japan. And third, so from the LCMS chromatogram, so please pick the one target peak. In this case, the observed MPZ of the target peak was 710.76. Using the molecular weight, please search the natural product database. In this case, so NPedia, so natural product database was used. And we searched within the weight of plus minus within a tolerance of plus minus 0.5 Dalton. And as a result, 11 candidate compounds were obtained. And next, we applied the retention order prediction model to these candidate compounds. For these candidates, the model predicted their relative retention position with respect to the 22 reference compounds. And this is the result of retention order predictions. And the match score indicates the agreement between the prediction retention order and actual retention order. So therefore, higher match score indicate better agreement between prediction and the actual order. So as a result, 4 candidates showed high match scores. So among them, so white one was jaspamide. This is previously reported from jaspisplendens. And fortunately, the MSMA spectrum of jaspamide has been reported in the previous paper. And comparison with our experimental MSMA spectrum showed strong agreement with the reported one, and allowing us to identify the target compound as jaspamide. So overall, this result demonstrates that retention order prediction can effectively support the PNPA identification. It's narrowing down from 11 candidate compounds to 4 candidate compounds. And this is a summary. We developed a retention order prediction model for PNPAs by integrating peptides and small molecule datasets. The model achieved an accuracy of over 0.9 and effectively supported the identification of PNPA with limited MSMS reference data. Importantly, we demonstrated its practical utility using real response samples. And the model codes are publicly available on GitHub here. So, and thank you for listening.
Thank you, Shohei, for this nice talk and really interesting. For me, it was interesting when you showed that combine the two data sets, the peptide and molecule, you have a better, a higher accuracy. And we have a few questions in the Q&A. So the first one, it's do you see rotation order prediction models being useful as soft constraints to support peak alignment and metabolite annotation in target LCMS workflows?
Okay, sorry, do you see the
Can you see the question?
yes, I can see. You see Sorry. Sorry. So, yes. In this case, yeah, I aim to use in the untargeted LCMS, untargeted metabolomics. Yes. So that's all. Sorry.
Another question here was, what was the range of retention times trained to the model, and does it work with retention times outside of that range?
Yeah. So sorry. Sorry. So, sorry. what was the range of retention time training? Yeah. So in training model, so retention times is many variation. It depends on the data set. For example, I use a 4 dataset from Predicate database. The first one dataset is from 0 to 10 retention time, and another dataset is 0 to 50 retention time. Do it works with this? So
There is another.
yeah, so that's this. So this model is not dependent on the retention time range. Yeah. Sorry. Where is the second question? Sorry. So where is the third question?
Yeah.
So which fingerprint did you use to predict relation? So mainly I use so I also use other fingerprints. I mix the 4 types of fingerprint: max fingerprint and circular fingerprint and other.
Okay.
And so many questions. Thank you. So where should we? Professor Arita, thank you for answering the question.
Maybe one more from me. How transferable is the model to different, say, like LC conditions, whether that's the gradient or the chromatographic conditions?
Sorry, where is that question?
Oh, that's just from me.
For you. Okay. So, please say one more time. Sorry.
So how transferable is the model to different LC conditions, like different solvents or gradients?
So it depends on if the reverse if you use reverse phase conditions, you can use my prediction model. And even if the gradient pattern is not the same. Yeah. Sorry. So, but linear condition is the better, I think. Yeah.
Thank you.
So thank you, Shohei. I think you have no questions. So thank you for this talk, and we can introduce Eisuke.
Yeah.
Great. So our next speaker is Dr. Eisuke Hayakawa, who has a PhD in general science and is an associate professor at the Kyushu Institute of Technology in Japan. With his expertise in metabolomics, he developed a platform to reanalyze and compare public metabolomic data, which he'll show us today. And his talk is titled Towards Repository Level Reuse of Metabolomics Data Through Metabolic Changes. So thank you, Eisuke. Over to you.
Okay, thank you. Thank you for the introduction. So let me share my slide. So is it working now?
Yeah,
that's great.
that's perfect.
Fine. Okay. Okay, yeah. Thank you for giving me this great opportunity to present my recent project. So I'm from Kyushu Institute of Technology and also a member of RIKEN CSRS. And my research focuses on integration of mass spectrometry with informatics to analyze large-scale metabolomics data. Today, I want to talk about challenges in using metabolomics repository data, large-scale data. And I also will present our project, a platform designed to enable repository-level reuse of public metabolomics data. So public metabolomics repositories have grown rapidly over the past decade. We now have thousands of studies across diverse biological systems, disease, and experimental designs. So this represents an enormous opportunity for us to using such huge, gigantic data sets. But also it means a major challenge because simply having more data doesn't necessarily mean that we can reuse it effectively. So this slide, I want to explain the core problem, I think. In metabolomics repositories, data are typically provided as mass spectrometry raw data and feature table consisting of the name of metabolites and samples. These feature tables contain signal intensities, and they strongly depend on the analytical platform used, such as separation method, LC method, and type of mass spectrometer, and ionization method, and many other acquisition settings. As a result, values in the feature tables, they are only meaningful within each study, and there is no shared scale across studies. So I think this makes a direct cross-study comparison almost impossible. Despite these challenges, we wanted to make large-scale reuse of public metabolomics data possible. To achieve this, our goal is to unlock the information stored in repositories by transforming the data into the form that can be compared across studies. The question is, how can we compare studies across different platforms? Absolute signal values in feature table, signal intensities in the feature table, they are not comparable across studies because each study has different analytical platform. So the values, signal intensities of feature table, they are only meaningful in each study. In contrast, relative changes, for example, like controls versus treatment, and disease and disease versus healthy sample. Such differential profile, they are much more robust. The direction and relative magnitude of the change, they are often conserved, even when measurement condition is different. This observation motivates our focus on differential profiles. Differential profiles summarize how metabolites change between two conditions, and it's expressed as fold changes for each metabolite. Once feature tables are converted into fold changes, all studies share same basic structure. A list of metabolites with relative increase or decrease. This can be used as a standardized and comparable unit across repositories. We systematically generated differential profiles at repository scale, currently using metabolites and the Metabolomics workbench. We processed over 2,200 studies covering various metabolites. From the resulting differential profiles, we obtained millions of fold change values, whose distribution is shown here. So this illustrates the scale and coverage of differential profiles derived from repository data. Importantly, all these profiles share the same internal structure, and they can be treated as referenceable units. Once we have differential profiles, now we can compare them directly. To compare differential profiles, we are currently using a method called IDMET. This IDMET approach quantifies similarity between two differential profiles based on the shared up or down regulated metabolites. This similarity relationship naturally extend into a graph structure. So now here you can see the gigantic graph where the nodes represents differential profiles and edge represents a metabolic similarity. Metabolic similarity alone only tells us that the studies are similar in terms of metabolic change, but it does not explain why they are similar. To interpret such similarity in metabolic change, we also need biological and experimental context. To address this, we systematically and automatically extracted structured metadata from study text contained in each studies in repositories. This allows us to associate each study with standardized mesh terms for disease, genes, and chemicals. So far, I have introduced differential profiles and similarity-based relationships and structured metadata as separated components. Currently, we are building a platform called IntegMet that integrates these elements into a knowledge graph-like structure. In IntegMet, Differential profile describes metabolic changes, while metadata provides biological and experimental context. By connecting studies and differential profiles and metadata, we create integrated structure in which these elements are systematically linked to each other. This platform IntegMet can be understood from three key aspects. First, it provides large-scale collection of differential profiles derived from public metabolomics repositories. Second, it supports visualization of similarity relationship of studies across in repository scale based on metabolic changes. Third, it serves as a knowledge base, linking metabolic change information with biological and experimental metadata. Together, these aspects enable cross-study exploration of metabolomics at the level of metabolic change. One way to use this platform is to obtain a repository scale overview of metabolic changes by combining metadata and metabolic change information. In this slide, for example, by filtering studies using keyword, disease-related keyword, such as inflammation or cancer-related keyword, we can survey similarity of differential profiles associated with those phenomena across repository. And also community detection then indicate clusters of differential profile that share similar metabolic change pattern across studies. This platform, IntegMet, enables layered analysis starting from a broad repository scale overview. We can first survey metabolic change pattern associated with particular phenomena or disease of your interest, and identify clusters of differential profiles that shows a similar pattern of metabolic change. Because differential profiles, as well as edge of metabolic similarity, they have metabolite level information. So you can then zoom in particular clusters, then identify consistency of altered metabolites. And you can use such information of altered metabolites, then perform more in-depth analysis, such as enrichment analysis. Particularly speaking, we are developing a web-based interface for this IntegMet. And it allows users to search for studies by metabolic change patterns, also find studies with similar differential profiles. And it can be also used to extract condition or tissue-specific subnetworks in repository level. And also it can overlay metadata to interpret why such studies or differential profiles are similar. It is designed as an exploration tool for public metabolomics data. So in summary, direct comparison of raw metabolomics data across studies is quite difficult, and which limited effectiveness of reusing public metabolomics repository data. This new platform, IntegMet, addresses this challenge by representing studies as differential profiles, integrating metabolic change similarity and together with metadata. As a result, users can explore metabolomics data at repository scale, and moving from global patterns to detailed metabolic interpretation. This platform Integrate is currently under development and available in as a closed beta. If you are interested in using this platform or following its progress, you can check our update on the SNS such as X or Blue Sky. So yeah, this project is a result of close collaboration across multiple institutions. And I would like to thank all collaborators. Yeah, I think that's it.
Thank you, Professor, for this nice talk and really interesting. And do we have any questions? And I have a curiosity. Can you tell a little bit more about how does the tool handle with missing data, the missing values?
Missing values
Yeah.
you mean missing values in feature table?
Yeah.
Well, actually, that is very technical
Okay.
question. But normally, I don't. We don't really. If particular metabolites showing a missing value, then we can just simply remove the column.
I think we have a new question. What do you need to do as a metabolomics community to help these types of databases grow or improve?
You mean repository or this kind of new platform?
Maybe both, I don't know.
Well, I think it's really important to improve description, a detailed description, as well as tagging with correct keywords. Because when we started this project, we first checked the keywords registered in each repository. But in most cases, users doesn't necessarily put enough keywords. Many studies just has keywords such as LCMS, and that's it. That's why we had to curate. We had to curate metadata by ourselves. So I think for the community, I think it's really important to annotate metadata properly.
Thank you.
How do you deal with conflicting annotations between studies?
Sorry, completing annotations?
Conflicting, like disagreeing annotations of, say, metabolites.
Currently, we are using feature table, so we are relying on the annotations by the users, by the researchers. But also, we noticed that but many studies, the annotation is not really reliable, I have to say. Like annotation is based on the exact mass, and that's it. And some studies, annotation, the procedure of annotation is not 100% reliable. So yeah, then now currently we are reviewing how the annotation is performed in each studies.
Great, thank you. There's a question here asking if you could please explain that the features are platform-based. Does that mean we should compare samples with as many platforms as possible?
Platforms, you mean analytical platforms?
Yeah, I think so.
So the question is, I'm not quite following the question. So the platform difference. So
Many
can I see
softwares. Yeah, it's written down.
Okay. Could you please explain the features platform-based? Does this mean that you should compare sample with as much platform as possible? Like, let me see does that mean that we should compare sample with as much? Well, our approach convert mass signal intensity into fold change. So in theory, I mean a fold change or up or down regulated direction of each of metabolites. So that means that even if the studies from different analytical platform, they can be compared. That is philosophy or that's the idea. So I'm sorry, maybe I'm not quite following the question, but
I think it's, yeah, that makes sense to me. So if we're using this, you would use the raw data, or would you use a like QC corrected data like input?
we are not using raw data because converting raw data into a feature table or the data matrix itself is a huge task. So we are now just using a feature table.
Yeah, sorry. I mean, so you have your feature table, and then often you know how you would process it or filter it based on your exclusion parameters. But so would you use the sort of raw numbers feature table, or would you use your normalized and scaled, say, feature table?
We are currently testing several methods, but as you know, not always normalization doesn't always work.
Yeah.
So we are now comparing which method is more robust or not. But actually, the main challenge actually is that a sample number for each group, sample number for groups is often case quite limited. Like some studies only shows 2 or 3 samples per conditions, something like that. Before starting this project, I was thinking like at least 5 samples for each condition is minimum, but in reality, many studies, the sample number is quite limited, and so that is rather than normalization method, number of sample for each condition is more critical. So
Yeah, I think we need to get the basics down first, the metadata and the sample number first before we move on to other things, right?
So put more sample number for each condition.
We have a new question, Luke. Thanks for the presentation. I have one question. What is the main difference of your platform versus GNPS type platform?
GNPS type platform, I'm not quite following what that means. So, I mean, GNPS is like spectral library for natural product or spectral network. So I think maybe maybe could you please elaborate the question a bit? So maybe I'm thinking like GNPS has, the molecular networking is like graph representation, but obviously molecular networking node is for spectrum. So our method node is differential profile or study. So am I answering question?
Maybe it's related to the node connection, the cousin, and I don't know.
Yeah, GNPS. I think GNPS molecular networking is for spectral level graph, but our platform is study level graph. So that's the difference.
Okay. So I think we have finished with the questions. And first, I would like to thank you again, Professor Masanori, you, Professor Eisuke, and Shohei, for accepting the invitation and for doing this at such time night for you. We truly appreciate it. And before we finish, I would like to share the next webinar. So I will share my screen. Can you see it?
Yeah.
So on behalf of the EMN, we also would like to invite you for the next webinar coming up in February, which will be on the Metabolomics approach applied to the brain disorders. And the webinar will take place on February 26th, and we will be joined by Professor Alessandra Sussolini from the University of Campinas, Unicamp in Brazil, and also the Dr. Henrique Caracho Ribeiro from the Orego University. And we always look forward to seeing you there. So thank you all for joining us today. We truly appreciate your time and engagement. And also, you can stay up to date of the upcoming webinars and the EMN activities. So you can follow us on the social media. We are on Blue Sky, LinkedIn, and also X. And if you want to, you can also contact us via email. So thank you. I don't know if Professor and Breanna want to say something, but that's it for today.
Yeah, all good here. Thank you, everyone.
Thank you. Bye-bye.
Bye.
See you.